Welcome to lab number 1 for Biology 100. This lab is a little bit special. You won't get a video introduction for all of the labs. And the reason for that is that our lab manual is pretty self-explanatory. It's designed for a student to walk in, sit down, and do everything on their own. Now, that's not how things work in real life. In real life, you do have an instructor explaining things, working alongside you. But for our online labs, I will not be doing a video for every lab. There are a few where I do make an introductory video or a video of some specific parts of the lab, or we link to reference videos for you to get an idea of what's going on. But lab number 1 is a little bit different even in our in-person labs in that we work together as a big class and we design an experiment as a big class. We brainstorm, we discuss all of the different parts of the lab. So basically it is very instructor-led, which is unusual compared to the other labs. So I want you to get some of that support as well. So I did record a video for lab number 1. Again, you won't get them for all of the labs. You won't get it for most of the labs, but there are a few videos here and there, especially supplemental. So in lab number 1, we're going through the process of science, and the majority of your lecture videos talked about the process of science. In other words, the scientific method. How do we answer the questions about the world around us? So the scientific method attempts to provide an explanation. You probably have already realized that you actually do this all the time, and I believe I mentioned this in my lecture video. Every time you come across a problem, make an observation, you try to explain why something has happened. And that's exactly what the scientific method does, although it follows a few specific steps to guide the researcher to make sure that they're following these steps so that they can create a testable hypothesis, produce an experiment that actually tests the hypothesis that they are testing, and then makes an experiment that is sound and gives you scientifically backed results. Now, that does not mean that you're always going to prove your hypothesis correct. In fact, many times hypotheses prove to be incorrect, but many times you will support your hypothesis, and that's a good thing as well. For this lab, you are going to want to open a few things from our Blackboard course. Obviously, this video. There's also a video on how to do the graph at the end. There's a data file with the data for our fish experiment. There's also a video that actually shows the experiment taking place. For this video, I'm gonna kind of bounce back and forth between giving you some information, letting you figure it out. So you might need to pause this video, answer some questions, go read something, and come back. So just use this however you would like. You are certainly welcome to go through it all on your own. I wouldn't suggest that because I'm offering help, but that is totally up to you. So the first step in the scientific method is to recognize that a problem exists or to make an observation about the world around you. So for now, I would like you to pause the video, go back, read the background section, and then in your summary sheet, state the observation that we're making as a class. So this is something that I would talk about in class. We would look at a fish and we would make this observation as a class. Well, we're giving it to you, so go ahead and go find that. The next thing that you're going to do is ask a causal question that attempts to explain the reason for the problem or the observation. Now, there are two kinds of questions, causal and descriptive. A causal question seeks to explain a phenomenon, where a descriptive question attempts to describe a phenomenon. So essentially, you're looking for the why something happens versus what happened. So I might ask, why is the sky blue? That's a causal question. A descriptive question about the sky might be, what color is the sky? Why is the sky gray today? Are there clouds present? Causal versus descriptive. So I would like you to write 2 or 3 causal questions that attempt to explain the observation in step 1. To get full credit here, you need to write down at least 2 causal questions. So pause this, write your causal questions, and then I'm going to give you the causal question that we're answering. Hopefully you've written down your 2 causal questions. As a group, you are a group even if you are online away from each other, the causal question that we are asking this week is, why do fish change the rate of operculum movement? So why do they change the rate that they move their mouths and their gills? So go ahead and write that causal question, circle it. So I want to see at least 3 causal questions, the 2 that you came up with and this one. If you happen to have already come up with this one, that's okay. Just rewrite it and circle it so that I know that the other 2 were yours. Now we are going to formulate a hypothesis. So remember, a hypothesis is not an educated guess. Okay, a hypothesis is an attempt to explain the answer to your question, or an attempt to answer your question using the background knowledge that you have about the subject. Now, I'm guessing that most of you are not fish experts, nor are you experts in respiratory movements of fish, and that's okay. We do give you some background information about the fish and why it moves its mouth. And we also give you the question that you are trying to answer. So I want you to formulate a hypothesis based on the given information. Now, I want you to list 3 hypotheses. Keep in mind that you should not have specific data in your hypothesis. So for example, if I were to try to ask a question and answer it with a hypothesis, I'm not going to include any information about my experiment per se. I don't want to see any numbers or any measurements in your hypothesis, just a statement that is attempting to answer the question. So I want you to write down 3 hypotheses and then choose one. The caveat here is that you might choose one that does not answer the question, does not fit with the test that we are going to be making today. So I want you to choose one, circle it, and then when I go through the test, I want you to put a little star or otherwise mark the hypothesis that actually fits the experiment that we're doing the most closely. And if none of them work, don't erase them, just write a new hypothesis that does work with the experiment. So I want to see all of your thought process here. So don't erase, there's no right or wrong answers per se in some of these. I just want to see what you're doing. Next, we are going to design our test. If we were in person, we would do this as a class, and I would give you all some hints using the equipment that we have available. I'm going to generally outline it, and you are going to think through the steps that you would use to actually do this experiment. So we're going to do this experiment by taking a goldfish and slowly lowering the water temperature and then slowly increasing the water temperature, and we're going to count his operculum movements as this happens. So to decrease the water temperature, we're going to take our friend Cheeto the fish, we're going to put him in a beaker, and we're going to set that beaker into a tray of hot water, which is going to slowly raise the temperature. To lower the temperature, we're going to slowly add ice cubes to Cheeto's beaker. Make sure you take him out of the hot water, otherwise you're being counterproductive. And as we raise or lower the temperature in 3 temperature increments, so every time we hit a new temperature 3 degrees from the previous one, we are going to count operculum movements for 1 minute. So as part of designing a test, you do want to write out the step-by-step instructions so that somebody could repeat your test. And you're going to do that in section 4F. But section 4 also asks you a few other things about your test. It asks you to identify the independent variable and the dependent variable. So the independent variable is the factor that you change. It's the thing that you have control over. So in this case, you need to think about what factor in this experiment are you changing, are you controlling that might affect the fish's operculum movement. Then the dependent variable is the data that changes as your independent variable changes. So you'll have to think about what data are you collecting that is dependent on the independent variable you're changing. There are two other important things to think about in your experimental design. One is the control group. Now, sometimes a control group is appropriate. Most times a control group is appropriate. Remember that a control group is somebody or a group of somebodies, a group of situations, not situations, a group of organisms, or in this case fish, that are not receiving any kind of treatment. Sometimes your control is actually a particular part of your experiment or the normal part of this fish's daily routine. So think about whether or not we have a control group or if we just have a sort of control situation, some part of this experiment that would be the normal or the no treatment part of this experiment. The other important thing in experimental design are the controlled variables. Now, this is different from the controlled group. The controlled variables are the outside things in the environment where you are performing your experiment, or things that happen in the experiment as it's going on that might affect the outcome. Now, ideally, you want one thing to affect the outcome, and that's your independent variable. But sometimes there are uncontrolled things in the environment around your experiment that you cannot control for. Now, the key thing here is that you want to control for as many things as you can. You want to be as consistent as possible throughout your experiment to keep those controlled variables at a minimum. So, for example, we are using goldfish today. Let's say we all decided to use different fish. That's an uncontrolled variable because you cannot say that your independent variable that you are changing is the effect, is causing the effect on the operculum, or if the fact that the fish are different is causing the change in operculum movement. So you want to think about in your experiment any outside factors that might affect the outcome, and you want to minimize them as best you can. These are really important in affecting your confidence in the data. Now, you are going to have to address confidence in your conclusion, and you're going to have to address confidence in the application questions. Experimental controls or controlled variables should increase your confidence. The more things you can control and eliminate as possible factors in affecting your data, the more confidence you have. Sometimes you cannot control all the variables. Sometimes you choose not to. And there are different reasons for this, but you want to minimize those uncontrolled variables. So for example, if we can control what type of fish we use, we want to use all the same type of fish. That's going to remove an experimental variable. Any uncontrolled variables should decrease your confidence. The more uncontrolled variables you have, the less confidence you can have in your results. The biggest mistake students make here is they'll say, well, I have this uncontrolled variable, but I don't really think it's important. Okay, it doesn't matter if you think it's unimportant or just that you know it exists. The fact is that those variables are uncontrolled and you have no idea how they are affecting your outcome. So even if you don't think they're important or you just know they exist, it does not eliminate them as uncontrolled variables and does not eliminate them as important. So keep this in mind when you're doing your application questions, that the more uncontrolled variables there are, the less confidence you can have that you have supported your hypothesis, even if you don't personally think they're a big deal, because you don't actually know how big of a deal they are. So for now, go ahead and answer step 4 questions, designing your experiment. The next thing we need to do is make a prediction. So I want you to review step 5 in the introduction. The basic format is if your hypothesis, then you predict expected data and results. So there should be no data and results in your hypothesis. Your data should be in your, in your prediction section. Now, one thing students do is make a circular argument here. Let's say that I want to know why the light isn't on in my office. My hypothesis is the light is burned out. My prediction would be the light bulb is burned out. My prediction might be if my hypothesis is, I suddenly am having a little brain issue. If the light won't turn on, my prediction is changing the light bulb will cause the light to work. Sorry. If my light won't come on, then changing the light bulb will make it come on. Okay, so you want to have some sort of specific data. So for this, you're going to say state your hypothesis about the fish, then increasing the temperature will do what to the operculum movement? If your hypothesis, then increase or decrease the temperature will cause increase or decrease in operculum movement. With a hypothesis, there is not necessarily a right or wrong answer. When I grade a hypothesis, I'm looking for a couple of things. Do you have a clear understanding of the background information? And therefore, does your hypothesis make sense with the background information? It doesn't have to be correct, but I want it to make sense with the background information. So occasionally students will say, well, I made a hypothesis, it's my guess. And I say, well, it does not appear that your hypothesis has anything to do with the question or the background information, in which case you will lose points. For the prediction, I am looking at whether or not the prediction makes sense with the hypothesis. So going back to the light bulb, you say, if my light is burned out and I change my light bulb, then the dishwasher will go on. Well, that doesn't make any sense, okay? So you want your hypothesis and your prediction to sort of match and make sense. So if you say something's going to increase, your results should show an increase. Or you might say they're inversely proportionate or whatever, but they do need to make sense with one another. So generally, as long as these make sense and are written in the correct form, you will get credit. So you're ready to perform your experiment now. If you need to make edits to your experimental procedure as you go, just write them in a different color pen or make a little star next to anything you edit. Again, I don't want you to erase and rewrite it. I want to see what your process was, and you can edit it, but just keep your original ideas. You are going to watch the Cheeto the Fish video that's linked on Blackboard Lab number 1. This is going to go through our procedure of raising and lowering the temperature and counting operculum movements of Cheeto. And then you will need to download the class data file. You'll need to add in the temperature increments that we measure for Cheeto. There are also 2 additional videos linked in the Blackboard Lab folder. One is my video on how to graph your results. The other one is another instructor from somewhere, not SWIC, that has assigned this lab to her class, and she sort of goes through the same things that I've just done here, but talks about a few other things. So if you are still confused, you're like, man, I don't understand what this lady's talking about, you can go watch her video and hopefully that'll clear things up a little bit. You do not have to watch that other fish respiratory video unless you want to. So you're going to perform your experiment, you're going to write down your results, you're going to follow the instructions in your lab manual, and you're going to calculate the average respiratory movement for each temperature, and then you are going to graph 2 lines. I should only see 2 lines on your graph. One is your fish's or Cheeto's operculum movements. The other is the class average, which should include Cheeto's movements. And again, you can watch the video if you need help there. All right, so step 7 is the conclusion section. Please review that in your lab manual, and you're going to make a statement about your results. Here, I don't really ask you in your conclusion statement to make any comments about your experimental procedure. Sometimes I will say, hey, I want you to explain whether or not this makes sense. Did anything weird happen? Were there any problems in your experiment? How confident are you? But for today, you're just going to state my hypothesis supported. I'm sorry, my data supported my hypothesis, or my data did not support my hypothesis. This is also the place where you would talk about those uncontrolled variables. Did you have anything that you couldn't control, and how might those have affected the experiment and your confidence in your results?